James, thanks for being here, and we really have a great pleasure to have you here.
Yeah, thanks for having me.
Yeah, so I heard you had a great talk today, and your research is mainly focused on the PET ligand, especially for the Parkinson's disease and other related neurodegenerative disease. And as a neuroscientist in the pharma, I really understand how challenging and important it is to develop a PET tracer for the patient stratification and also drug responses. And what motivates you to start this field, and what's your thought when you initiate this project?
Sure. So my lab had been studying alpha-synuclein misfolding for a number of years from kind of a basic biochemistry and biophysical point of view. And then speaking with people that were on the more clinical and translational side at UPenn sort of made us realize that we could make contributions that could help to develop these molecules. And so honestly, it was actually some students in my group that really wanted to kind of push the translational side of things, and so spurred me to start thinking about these things. And when we got in there, we realized that we could make some valuable contributions.
Yeah, it is really important, and also I think like the PET tracer development is more and more important in general in terms to have a successful clinical trials, right? But as far as I know, like for example, the A-beta and the tau PET tracer has been widely used in this field for maybe more than decades, right?
Yeah.
But in terms of Parkinson's disease alpha-synuclein PET tracer, there's not too many. I know there are some clinical trials ongoing, but what's the technical and scientific challenge to develop a PET tracer for the Parkinson's disease?
Yeah, so one of them is that the ones for Alzheimer's, they target the A-beta or tau, and those are just much more abundant aggregates than the level of alpha-synuclein is in Parkinson's. And so I think there's just a pure question of how much target is available. I think there are some aspects of the misfolding of alpha-synuclein that makes it challenging, but I also think that people are appreciating more and more the different forms of tau in not just Alzheimer's, but things like chronic traumatic encephalopathy. And so there's sort of new challenges emerging as people try to develop PET probes for those diseases as well.
Yeah, I know like a lot of companies, like including Merck, we are working together with, like, for example, Max J. Fox Foundation and also some academia lab. And from your perspective, how can we facilitate the collaboration between academia labs and also pharma industries and to push the PET ligand development?
Yeah, that's a great question. I think, obviously there's certain restrictions on sort of how much data can be shared. I think, as we sort of, for us as academic labs, our goal is to publish sort of all of our SIR data. And so in that direction, we can certainly share that and help industry collaborators. I think within IP restrictions, sharing biosamples and biomarker things like that can help to stratify what we use in our studies could be something that could be easily shared without so many IP restrictions around constructions of matter and things like that for ligands.
Yeah, it is super important. And for this specific conference, and our theme is like AI and the new modality in the drug discovery. And do you think the artificial intelligence can potentially help the development of PET tracer in your field, especially, for example, the ligand design or the imaging analysis? Have you seen any implication for the AI in your field?
Yeah, certainly. We do some machine learning and AI work around ligand SAR, and there's definitely a role that can be played there. I know that the people on the clinical end are also using new methods for analyzing the images. That's not my area of expertise, but I think both ends of those things, as well as as you start to get large patient cohorts, maybe analysis of patient background information in ways that are not obvious to humans could also be of value there. So. Yeah,
definitely. We do see more and more application of AI in the drug discovery in general. Yeah, I'm glad to have you here, and look forward to see you around at the conference.
Yeah, thank you for inviting me.
Thank you.